protein complex involvement of microprotein was discovered. So these microproteins A and B, they interact physically, as you can see from these two hybrid and the confocal images, and pull down. So the physical interaction is there, the evidences are there, and there are evidences that it forms or participate in a bigger complex. It's not anymore a dynamic situation, and the flowering is one of the most complexly regulated trait in plants, and that's how entry of this microprotein MIB1A and MIB1B make it more interesting to learn how complex traits or complex quality or physical traits in plants are getting regulated. So we talked till now about transcription factors and regulators. Can we also use microproteins for diverse background of proteins or regulator proteins? So that's the question the lab asked, and all microproteins belongs to the protein classes. So this protein identification appears into an approach called synthetic microprotein. They are naturally occurring microproteins to regulate transcription factor, and they are well known, as I described in previous slide. But is there any way we can control diverse proteins using microprotein as a tool to control the protein functionality? And this paper in plant phase demonstrate how you can design microprotein, a synthetic microprotein, and target your protein of interest to use as a tool. So Ula, my colleague, it was her PhD project, and she identified that the microprotein percentage in Arabidopsis, it represents a lot hydrolases, as well as other key regulator enzymes, the receptors and lyases. So she choose one candidate from each class, dicer-like protein, which belongs to hydrolases, bri1, which is receptor, and then cry1, which is a lyase. So I will here discuss how one of these cry1, one of these three, and I will focus on cry1 study, how they define, how the lab define how to design microprotein and how you should validate that. So cry1 is a protein with 3 different domains. She designed a MIP cry1 from pH domain. How this works, whenever there is a blue light, cry1 opens, and that causes deacetylation in seedling. If you overexpress a microprotein cry1 and expose it to the plant to blue light, there is a etylation. It means that MIP cry1 is causing non-dimerization of cry1 and resulted into better or less reception of blue light, causing etylation or elongation of seedling during early germination days. So again, validation part includes yeast to hybrid interaction. So she validated. And then, as you can see here, this is col0 wild type, cry1 seeds, mutant seeds. So in blue light, all MIP1 overexpressors, they mimic phenotype of cry1 mutant. It shows that whenever you overexpress a cry1 MIP, it suppresses functioning of cry1 protein at post-translational level and causing a phenotype which is similar to a mutant in wild type, dark as well as blue. So we discussed these approaches, how to use it as a tool, but we also wish to make it more available to public, and we wrote this paper about how to use microproteins and their approaches to build a tool in biotechnology. And we talk here about 2 different approaches, and this also evolved with time. Earlier, we were only talking about approaches where there was a repressor complex was forming. That means microprotein was binding to a protein and then repressing its function. But we also propose here that a protein can also get activated by binding to microprotein. And that was one of the case that was reported in plant physiology paper. So we propose this pipeline, how to identify microproteins, and then filter the information based on MIPFinder information pipeline or software, then target identification for microproteins those you are interested in, or this could also go reverse way. You can identify first your target or a protein with multiple protein complexes which you find really interesting, and then you can use or design a microprotein to control that. Or there are ways you can use synthetic microprotein, or you can use MIPFinder to identify if there is an interesting microprotein like sequence available in genome that could target my protein of interest. And then you validate your protein microprotein complex by biological function, for biological function using experiments that I defined before. And we will come to that part again in this end. So as a biotechnological tool, this paper from Stephen's lab defined first time how to use microprotein as a biotechnological control. So we design a microprotein from B-box2 HD MIP CDS from a transcription factor, which is HD1, a major master regulator of flowering in rice. And why we need to do that? So the biotechnological intervention, they always have a question and a need to answer. What was need here? In Nordic countries, especially Denmark, Finland, Norway, Sweden, the duration of days during summer is really long. It's like in Norway, there is always sun, as several of us know already. Even in Denmark, the duration of light is really long. We call them strong, long days. So strong, long days as well as short days, they inhibit the flowering time in plants. And this sense of short, long days or short days or strong, long days, it's sensed by machinery of transcription factors in plant. And that's how you signal plant to flower or not to flower. But this reception should be mediated by a better control that reflect how plant should respond to lighting condition outside. And if there is a flowering, there is a yield. Better flowering, better yield, of course. So to cover up the situation of strong, long days, what we design is an approach using the microprotein biotechnology to inhibit the HD1 transcription factor, HD1 transcription factor using HD MIP CDS, HD MIP HD1 microprotein. So you can see wild type during strong, long days don't flower, while the mutant of HD1 flowers pretty well. And when you overexpress HD1 MIP as a control, there is not too many flowers or no flowering at all. But if you look carefully, that HD MIP1, HD1 microprotein 2 overexpressing lines as well as one overexpressing line, they started flowering just as like almost as similar to the HD1 mutant. So, and if you look here, during short, long days, the flowering time, the heading time is flowering time. Flowering time has reduced a lot for HD1 mutant and the microprotein overexpressing and targeting HD1 in rice. So this tool behaves in a way that using a microprotein inhibition, you can control HD1, and during the extra long days, it cause a phenotype similar to short days and cause the flowering. So in a situation or a geographical situation like this, you can design your plant in a way so that you can control flowering time. But again, this is still limited to a particular trait the lab has been studying for a long flowering time. So we wish to move further deeper into the fact that if we are moving to crops, there are more challenges to address: stresses, pathogen attack, yield control, quality control. And we wish to expand this opportunity of using microprotein to control not just flowering, but also multiple diverse paths, metabolic engineering, for example. So we designed a project to identify microproteins in major crops, especially cereals. Cereals are being consumed across the globe very well. And we came up with this paper, which recently published in G3, Genetic Society of India, America. So what we did to refine our quality of microprotein identifying from crop genomes is we reshuffle certain things, we improve certain pipelines in MIPFinder. For example, the potential microprotein characteristic, we define that they should have a homologous interaction partner. They should contain single domain and high prediction instability index, which has been defined in our paper, how to use that. And then we made a filter of length less than or equal to 150 amino acid, should not have 10, more than 10 homologous in proteome, because plant genomes are really complex sometimes, thanks to polyploidy or the evolutionary recombination events. And then microproteins should exist on score less than 4 or equal to 4 on UniProt, because a lot of proteins we identified UniProt database. So we also put a filter on target. There are a lot of filters. Those were coming with the previous microprotein finder pipeline developed in lab. And then we improvised everything. So using the newest MIPFinder 2 pipeline or software, which is now available on GitHub. If you go through the paper and you want to use the software, it is openly accessible. You can run the command line very easily on your computer. So we identify proteins, microproteins, small proteins, single copy, and all other information that we need to engineer the crop plants. And we targeted barley, rice, wheat, maize, and sorghum. These are very famous cereals. Those are being consumed and grown for commercial as well as household applications or consumables across the globe. So these crop we investigated, they are really rich in microproteins. We segregated information based on small proteins. Those are smaller than certain size. And then using new filters, we define if these small proteins are microproteins or not. And you can see all the information here. For example, rice, which is one of the most consumed grain. It is really rich in microproteins, and there are diverse pathways these microproteins are covering. And certainly some microproteins are single copy. They make more confident cases if you want to study them using CRISPR or maybe other ways. And then the number of homologs they have. So let's go back to this proposal we put forward, how to study microprotein. So we invite everyone, you should go through paper and then follow up that has been done to use microproteins as a tool to study or design new crops or metabolic engineering. So again, there are certain things to understand. Alternative transcription, that also not limited to splicing, but also polyadenylation or early stop codon. Small ORF, how to identify these things. Then we have two different versions of MIPFinder. The modified one, which recently published in G3, was MIPFinder 2, openly accessible. Everyone should go and try that on genome of interest, not only on plant, but also on yeast or any living organism which has been sequenced, or you have the proteome information for that. Overexpress that and validate these things by protein-protein interaction assay, loss of function development by CRISPR or knockout or RNA, whatever you prefer. And then experimental analysis to validate if things that you designed are really working. And the information is not limited on to this. There are a number of other studies coming up soon from Stephen's lab, which I can't talk about here, unpublished, of course, but they will be out soon, and they will show how to use the microproteins to control or design metabolic engineering, or maybe quality trait development in crops, or regulation of certain things. So here I would like to stop and thank everyone. I would like to acknowledge Stefan for his consistent support during my stay, as well as even after that. He's always a good mentor. He's always mentoring, suggesting ways to improve science and general perception of science, in my opinion. And Valdoko, his new colleague in Stefan's lab. And I would like to acknowledge the past colleagues I had. Wonderful time with them. Anko, Bin, Daniel, Esther, uh, myself, of course, sorry, Shinyong, Tanai, and Wendy. And now I would like to invite you to ask questions if you have some.